this head trauma over many, many years of repetitive head impact starts some sort of process that leads to the accumulation of tau that we see in CTE. In some people, this process goes all the way to CTE and neurodegeneration, and in some people, it doesn't.
From the Harvard Kenneth C. Griffin Graduate School of Arts and Sciences, this is Colloquy, bringing the expertise of the university's PhD community to top-of-mind issues. I'm Paul Massari. It's a mystery that often begins on the playing field and ends deep inside the brain. Chronic traumatic encephalopathy, or CTE, is a neurodegenerative disease linked to repeated head injuries. It's been found in professional athletes, soldiers, and others who've experienced years of those traumas. New research from Harvard Griffin GSAS alumni Chanthia Ma and Guanlan may help us better understand this condition. Their study looks at the smallest units of brain biology, individual neurons, and finds surprising clues written in the DNA itself. Using single-cell genome sequencing, they discovered that neurons in people with CTE carry distinctive patterns of genetic damage, patterns that may overlap with those seen in Alzheimer's disease. Ma and Dong's work not only sheds light on how brain trauma leads to long-term decline, but also hints at possible shared mechanisms across different neurodegenerative conditions. With us to discuss CTE and the new research is Chanthia Ma. Ma graduated from Yale University with a Bachelor of Science degree in molecular, cellular, and developmental biology. She's currently an 8th-year MD-PhD candidate, finishing her MD at the Harvard-MIT Health, Science, and Technology program. Chanthia Ma, welcome to Colloquy.
Thank you.
So, for those who may have heard of CTE from stories about football players or boxers, what exactly is chronic traumatic encephalopathy? And how does it differ from other brain diseases like Alzheimer's?
Yeah, so CTE, or chronic traumatic encephalopathy, it's a neurodegenerative disease that develops after years, oftentimes up to 20 years, of mild repetitive head impact. The key difference between CTE and Alzheimer's disease is that even though they're both diseases of what we call the tauopathies, so they're both diseases of tau protein, the key difference is what causes them. So for CTE, we know the distinctive cause is these years of head impact. For Alzheimer's, for most people, we don't really know what is causing Alzheimer's disease. For Alzheimer's disease, 1% of individuals, it's a familial gene. For the general population, there are risk factors that we've studied. But for CTE, we can say that it seems likely it develops in the brain after years of mild repetitive head impact. The symptoms of CTE would include emotional difficulties, mood swings, short-term memory problems, issue with sleeping, headaches, depression. And as you get to more advanced symptoms, you would have executive dysfunction, trouble speaking, just a lot of issues with daily activities, even culminating in self-harm thoughts or behaviors. And I think that is a lot of where we see CTE come into the social, in the news and in the media, is seeing individuals who have these later stages of CTE where they're having issues with aggression and self-harm. But the diagnosis for CTE can only currently be made neuropathologically. For the pathology, and this is why there is that original link to Alzheimer's disease, is because CTE is diagnosed by looking at the accumulation of the tau protein in the brain. And we know that in Alzheimer's disease, there's tau, there's beta amyloid as well. And for the CTE individuals, especially in the later stages, there is a huge accumulation of this tau protein, especially in the sulci of the brain. And this is for those not super familiar with the anatomy of the brain, you would have the gyrus, which is the surface of the brain, as well as the sulcus, which is where the brain tissue invaginates into the sulci. And the tau in the early stages of CTE is found in the sulcus only. In the later stages of CTE, you actually find that the tau, as well as the markers of neurodegeneration, is spread to the whole cortex of the brain. And that is how our collaborators at BU, Dr. McKee, who's the expert on CTE, has been diagnosing CTE.
Your study looks at DNA mutations inside neurons, which most people think of as non-dividing stable cells. What led you to suspect that tiny changes in neuronal DNA might help explain why some people develop CTE after repeated head injuries and others don't?
Our lab, before I joined as an MD PhD student, has already been looking at somatic mutations, and it's correct that you say these are single DNA changes inside of individual cells. We looked at in aging brains as well as in diseases of DNA repair, and we found that in older brains, as well as in these other diseases, that there was a accumulation of these what we call somatic mutations. So these are mutations post-conception, so not a mutation you would get from a parent. In these non-dividing cells, which are the neurons of the brain. We found in our lab that as you age, there's an accumulation of somatic mutations in the brain, in aging, and in diseases of DNA repair. So this kind of links the idea of disease and lifespan and aging with the accumulation of somatic mutations. So that's why we're interested in looking at cells that are non-dividing, because even though they don't divide, they can still accumulate mutations in their DNA through different processes that we study in our papers and other similar labs.
You used a method called single-cell whole genome sequencing in your research. Can you walk us through, those of us who aren't neuroscientists, in simple terms, what that technique is, what it allows you to see, and why it's a breakthrough for studying diseases of the brain?
So we used single cell, which means individual cells, and whole genome sequencing, which means mapping out the DNA of the whole genome of that cell. And it's really groundbreaking for studying diseases of the brain because the neurons are non-dividing cells. So most of the time when we're thinking about studying the DNA, we actually take a lot of cells mixed together, like when we take a blood sample or swab our cheeks. But when we sequence all those cells together, it's actually just an average. And previously, we didn't have the technology or the power to be able to sequence individual cells. We need a large amount of DNA to be able to conduct the sequencing. So now with our novel techniques, we're able to sort for individual neurons, and we're able to amplify the DNA of those individual neurons to a level at which we're able to sequence the DNA. And then we're also able to sequence the DNA at a depth high enough, which requires a lot of computational power to get to the whole DNA sequence of individual cells. And that's a pretty new development, to be able to sequence at such a depth that we can map out exactly what mutations happen at a single cell level. Because previously, let's say you take cells from a different part of the body, like skin cells that divide rapidly or blood cells that divide rapidly, you actually gain a lot of information from sequencing those cells, because the cells that divide rapidly come from a progenitor cell, and when you sequence, let's say, a couple thousand of those cells, is giving you very similar information from the progenitor cell. But in neurons, because it's non-dividing, and you want to know the information in that individual cell, you need the technology to be able to, one, amplify it and to sequence it at a depth where you can confidently call the mutations from the rest of the body.
So can I ask you a basic CTE question then? You've mentioned the different stages of the disease and that it's progressive. If it takes place in cells that are by definition non-dividing, how does it spread?
I don't think our findings give us a definitive answer for that because we weren't looking at inflammatory cells of the brain like microglia and other immune response modulators. But we think that by looking at the somatic mutation accumulation, it means that something is going wrong in that part of the brain. So like I said, in earlier stages of CTE, the accumulation of these abnormal proteins, tau, in the brain only happens in the sulcus. But we see that as this disease progresses to the later stages, you see tau very much spread out across the rest of the brain, not just to where the originating force from head trauma happens. Oh, and I forgot to mention earlier, the reason that the tau accumulates in the sulcus is they did these studies looking at the physics of head trauma, and the accumulation of the most amount of force when you have repetitive head impact is in the sulcus. And that makes sense where there is the most accumulation of damage seen in the earlier stages of CTE.
You've discovered that people who experienced repetitive head impacts but didn't develop CTE didn't have those same DNA changes. So what does that tell you about the factors, whether they're genetic, environmental, or some other factors that might make some individuals more vulnerable?
So I want to preface that our study is actually the first single-cell genomic investigation into CTE, given it is a disease that is still being described and studied. So what it's looking like to us is that this head trauma over many, many years of repetitive head impact starts some sort of process that leads to the accumulation of tau that we see in CTE. In some people, this process goes all the way to CTE and neurodegeneration, and in some people, it doesn't. So the question is less so if there is something that is predisposing individuals to have CTE, and rather in the individuals that have CTE, some sort of neurodegenerative process occurred. So the somatic mutations are more so telling us it is a downstream effect from CTE rather than a causing effect of CTE, if that makes sense.
So what I hear you saying is because this is not something that's passed on from generation to generation, it's not like you can do a DNA screening for those who maybe shouldn't be playing football or shouldn't be boxing because they'd be predisposed to developing CTE.
Yeah, our study doesn't really show, actually our evidence isn't leading us towards there being some sort of gene that predisposes you to getting CTE, but rather we're hoping that our study reinforces that CTE is a real disease, it's linked with years of head trauma, and that these findings can hopefully influence activity choices of athletes, hopefully give us an idea of what switch happens when the brain goes from accumulating damage to developing neurodegeneration. Because if you actually look at the brains of these individuals in stage 1, stage 2 is just tau in the sulcus of the brain. As you move on to stage 3, stage 4, these are holistic markers of neurodegeneration. It's brain atrophy, you see a increase in the ventricle sizes, which is again a showcase of brain atrophy. And it's not just in one isolated area. So we're thinking some sort of inflammatory process perhaps kickstarts the neurodegenerative process that then makes CTE a neurodegenerative disease, similar to how Alzheimer's disease is a neurodegenerative disease as well.
Is there anything about this work, this study, that could eventually help doctors diagnose CTE in living patients? Does it still have to be done neuropathologically?
So there have been imaging studies done in CTE. There are a lot of imaging studies done in Alzheimer's disease right now. There are PET scans, PET tracers for amyloid. There are some tau scans done for Alzheimer's disease, but I think mostly in a research basis. I think similar studies could be done in CTE if once we develop our technologies where we can do somatic mutational testing, which requires a lot of data power currently to provide some diagnostic information, but that would need to be considered in combination with other symptoms of the disease, given that CTE is confirmed neuropathologically, a lot of times diagnosed with the clinical symptoms of mood changes, depression, and other executive function dysfunctions. So I think sampling brain tissue is very difficult in living patients, but looking at ways of accessing tissue like blood, where we can learn more about the brain, you can't find out information about somatic mutations of neurons from looking at the blood, but maybe other factors of inflammation could help us get an answer to CTE.
So building on the work that you've just done, what's the next step for understanding the process of neurodegeneration that takes place with CTE?
Yeah. For my study with CTE, we looked at single cell genomic changes in the brain. Our lab is also looking at other ways of studying CTE, but also other neurodegenerative diseases. And we have other studies that are coming out. They're in preprints now, where we looked at these signatures of oxidative stress, also the topoisomerase related deletions as well. And as we study that, we'll have a better idea of why those factors are linked with neurodegeneration. So as you know, topoisomerase is related to RNA transcription when the DNA twists itself in the wrong way. And earlier I discussed studies we've done in our lab where we looked at disease of DNA repair. So in these non-dividing cells, it looks like the ability to accumulate DNA damage is what is literally causing these somatic mutations and leading to an elevated number of somatic mutations, which seems to be linked with aging, seems to be linked with lifespan, seems to be linked with neurodegeneration. So some sort of modulator of these DNA repair mechanisms could potentially be translated into a clinical care, something that could boost our response to oxidative stress, either through medicine, lifestyle, diet. The drugs currently that target topoisomerase have a lot of side effects. They're mostly used in cancer, so probably not a good thing to test right now. But just kind of understanding that it seems, or the way that our body repairs DNA damage is very intimately linked with aging and disease, and specifically as I studied neurodegeneration.
From the Harvard Kenneth Griffin Graduate School of Arts and Sciences, this has been Colloquy. This podcast is an extension of the school magazine of the same name. You can find a transcript of the show, as well as past episodes, at the Harvard Griffin GSAS website, gsas.harvard.edu/colloquy-podcast. If you enjoyed this interview, please rate or like the show on Apple, Spotify, or YouTube so that others can find it as well. And be sure to follow us on those channels so you'll never miss a conversation. Our theme music is from the Boston-based band Francine. Check out their other tracks at www.francine.info. I'm Paul Massari. See you next time.